All right. Well, hey, folks, good evening. Welcome to another Noyo Center Science Talk. And this one, of course, is really exciting to me personally because I learned the word Leptospira when I started rescuing sea lions 20 years ago, pretty quickly. And that's how there was a sea lion that was sick that came in close to Little River, and that's how I learned about marine mammal rescue. That's how I then became a rescue person, and then became quickly more fascinated with what was wrong with these animals. So for me personally, this is just an exciting moment. And then right up to this year where we had a big uptick in sick and dead California sea lions, kind of a crunchy little spat of them in November. And so this, for me personally, it's really exciting for our stranding group up here. It's exciting both for live and dead. When I say live mammals, we do a lot of the Noyo Center volunteers and staff are actually Marine Mammal Center volunteers. So
okay, great.
we're wearing volunteer hats for Noyo Center and staff, and then so there's kind of a crossover there, but I do want to say that Noyo Center does not have any part of rescuing live mammals. We simply do as with our other hats on. So folks, you know the drill. Please do take the moment to support the Noyo Center for Marine Science. Great opportunity to go onto our website and become a member. I'm going to be pushing the membership just because it really helps us to be a sustaining member. And then you can come in and visit the Discovery Center Museum. And that's one of the benefits to being a member is you can come in for free. And additionally, you can get our Pinniped Press, which is getting a lot of feedback as being a really fabulous newsletter. So those two things are worth it alone. And you also support our efforts in bringing you these incredible scientists from all over the place who are giving up their time and teaching us more about the work that we're doing, diving a little deeper into some of the issues that we see. So support us, show us some love. So Katie Prager is a name that came in this little curiosity around the leptospirosis. And so our fabulous volunteers that seek science speakers for science talks reached out to her and she obliged. Lucky us, lucky Noyo Center to have someone so knowledgeable and who's had 25 years, was started out in Princeton and then came west, UC Davis and UCLA. And I just can't wait to dive in and listen to what she has to share with us this evening. I hand it over to Miss Katie Prager. Thank you. Thank you.
Great. Thanks, Sarah. Thanks for the really nice introduction. And for the opportunity to talk to all of you today. So today I'm going to tell you about the spirochete bacteria called Leptospira in the northeastern Pacific ecosystem. Primarily, I'm going to focus on Leptospira in California sea lions. But I will touch a little bit on the other marine mammals. I'll give you a bit of background, tell you about Leptospira and the sea lions, tell you a story about how it disappeared and came back, which was very exciting for us. And then, as I said, I'll touch a little bit on Leptospira in other marine mammals. Before I start, though, I just want to acknowledge that this work is only possible because of the tremendous effort from all of our collaborators. I am in Jamie Lloyd Smith's lab at UCLA and have been working with him since 2010. The Marine Mammal Center and the Marine Mammal Lab at NOAA have both been incredibly important collaborators. And then, as you see, there's a number of other people who have also been very important in getting this work done. So leptospirosis is the disease that's caused by infection by Leptospira, and Leptospira are spirochete bacteria that are pathogenic, and they're in the genus Leptospira. There are over 300 pathogenic serovars, and a serovar, you can think of it basically as is a strain of Leptospira. And the sea lions get Leptospira interrogans serovar pomona. But I'm just going to call it Lepto or Leptospira from now on because that's way too much of a mouthful. Leptospira in general is a globally important zoonosis, and a zoonosis, as many of you know by now, is a pathogen that can be transmitted from animals to humans, or non-human animals to humans. It can infect most, if not all, mammals. When an individual is infected, initially it will multiply in the blood, and then it'll end up colonizing various different tissues, but ultimately it colonizes the kidney, and then it's shed out in the urine. And so transmission is generally because of this urinary shedding. So there can be contact with the infectious leptospires in the urine, or in urine contaminated soil or water. And infected animals can range anywhere from having no apparent clinical signs to dying from this infection. And some animals can be infected persistently and be in this chronic carrier state where they shed the pathogen for months and sometimes even years. So when a sea lion becomes infected with Leptospira, the majority of the clinical signs are a function of the kidney failure that they're experiencing. So they might drink a lot of water. It's called polydipsia. Their mentation might be a bit depressed. They might seem lethargic, dehydrated. Their body condition is often fair to poor. They show gastrointestinal signs because of the ulcers, the GI ulcers that they can get. And so they can show signs of abdominal pain, like this sea lion here, who is in this kind of hunched position. They can have dark, tarry feces that are due to the digested blood from those GI ulcers. They might have diarrhea, vomiting, and they could also have oral ulcers. In live animals, or diagnosis is in live animals is through the typical clinical signs. At the Marine Mammal Center, generally it's based primarily on the clinical signs and then the serum chemistry results. So when the kidney function is disrupted, the serum chemistry results, the analytes that we test in those can be will look abnormal, and there's a kind of typical change to them that's very suggestive of leptospirosis. We can also take blood and look for antibodies against the pathogen. And then we can also look for the pathogen itself using PCR, culture, or something called immunohistochemistry, where you, and this is more in a dead animal, where you have special stains that allow you to detect the organism in the tissue. In dead animals, at necropsy, or which is basically, I'm sure you all know this, but it's like a autopsy, but on an animal. And so when you do a necropsy, you conduct gross pathology. So you look for any major changes to the kidneys. They can be, they tend to be pale and swollen on necropsy. And then when you cut them open, the typical definition that you see from the outside towards the inside is gone. When you do histopathology, so when you take really thin slices of those tissues and look at them under a microscope, you'll see very typical changes. And as I mentioned before, you can also stain those slices and look for the organism itself. Many of you, again, probably know this, but the of the sea lions that come to the Marine Mammal Center in Sausalito, sick with leptospirosis, roughly 2/3 of those animals die, despite the really excellent clinical care that they're given. But we suspect that a much higher proportion survive out in the wild, and we think this because when we look for antibodies against Leptospira in animals that have stranded for causes other than leptospirosis, we'll see really high levels of seroprevalence, or the seroprevalence is the proportion of animals that show antibodies in their blood. So a lot of animals will have these antibodies, even though they're coming in for other causes. And so that indicates that they were infected and they recovered from the infection. And then we also have detected animals that aren't sick but are shedding the pathogen. So this is subclinical shedding in free-ranging sea lions. So as I mentioned earlier, this work wouldn't be possible without all of our really wonderful collaborators. And really the biggest one is the Marine Mammal Center, which you're all familiar with up in the Bay Area, or down for you guys in Sausalito. And you can see the little arrow pointing to it. I don't know if you guys can see my arrow, but here. In this kind of bright green is the Marine Mammal Center stranding range, and then in orange is the range for in orange here along the west coast of the U.S. is the range of the California sea lion from the Channel Islands up to up to Alaska. So through our collaborations with the Marine Mammal Center, we have around 40 years of clinical data, and using these data, we can determine what the causes of stranding are of the various different animals that have come in. It allows us to characterize the outbreaks of Leptospira that have occurred. They've also shared a tremendous number of bank samples that have been collected over the decades. And then they've provided support for the field work that we do. The Marine Mammal Lab, which is located in Seattle, but their work takes them often down to the California Channel Islands, on San Miguel Island, San Nicolas Island, and San Clemente Island. Through their work, we have over 40 years of MARC resite data. And through these data, we've gained information on the California sea lion demography, so the population structure by age and sex, and then also survival by age and sex. We also have access through them to age and sex-specific movement patterns, which are really critical in providing insights into how the population is mixing. We also collaborate with the Oregon and Washington Department of Fish and Wildlife, and we have been sampling with them up in Astoria, which is at the base of the Columbia River, and then also at Bonneville and Willamette dams to get information from the animals that are up there. And those are primarily subadult and adult males. So when I started over a decade ago in 2010, we began a much more active collaboration with both the Marine Mammal Center and the Marine Mammal Lab, but also Oregon and Washington Department of Fish and Wildlife. And we began to collect data much more intensively. This included increased sampling and testing of both stranded and wild-caught sea lions, and increased diagnostic testing of both the banked and the prospectively collected samples. At the Marine Mammal Center, the vets, techs, and pathologists collected and sampled from over 5,000 animals. And with the Marine Mammal Center, the Marine Mammal Lab, and the Departments of Fish and Wildlife, Oregon and Washington, as well as many other collaborators, I led field efforts during which we captured, anesthetized, and sampled over 2,000 sea lions from 3 sites along the sea lion range. Oregon and Washington, which I mentioned, Astoria and Bonneville and Willamette, Año Nuevo, which is within the Marine Mammal Center stranding range, and then San Miguel Island, which is on the Channel Islands, which is where the majority of the sea lions have their pups. So the main rookeries are down in the Channel Islands. And from these animals, we collected blood, urine, vibrissa or whiskers, and in the stranded animals, we would collect kidney if they died, and we collected other measurements, morphometric measurements as well. I also, because we were sampling wild-caught animals, and it's not it does cause some amount of stress in those animals. I would always reach out to as many collaborators and other researchers that I knew of that were doing work on sea lions to find out if they needed any samples that we could collect easily without further stressing the animals, but by gaining as much information as possible from these individuals to hopefully reduce the amount of captures that have to happen ultimately in the population. The diagnostics that we would conduct on these samples were on the serum. We conducted something called a microscopic agglutination test, which is a way to detect the antibodies against Leptospira, and this gives us information on prior exposure and infection. We also collected serum for serum chemistry to assess their clinical status. In the live animals, we collected urine. In the animals that died, we would collect urine and or kidney. And those provided, those we would conduct PCR, culture, or immunohistochemistry, and that lets us know whether the individual was infected at that moment with Leptospira. And if we detected positives in the urine, that meant that they were shedding these infectious leptospires. And then from culture isolates, we were able to sequence the genome of the pathogen and then use these sequences to do phylogenetic analyses, which allow us to see kind of how related the different strains that we isolate are. So what did we learn from all of these data? This picture is from San Miguel. I learned that I really liked doing field work out there. We, using the long-term data set on sea lion stranding each month from leptospirosis, and that's what I have plotted here. You can see the x-axis along the bottom is time, and then the y-axis is the number of sea lions that stranded each month at the Marine Mammal Center and were diagnosed with leptospirosis. So looking at this long-term data set, you can see that there are yearly seasonal outbreaks that have been occurring since at least the mid-1980s, and that outbreaks peak in the fall, and that their magnitude or the size of these outbreaks varies, with really large outbreaks occurring every 3 to 5 years. Mathematical modeling work that we've done that was led by a postdoctoral researcher at UCLA named Benny Boremans. He has since moved on to private contracting. His work shows that the size of the outbreak is driven by the number of susceptible sea lions, so sea lions that have never been infected before by Leptospira, and especially by the younger male susceptibles, but also by certain oceanographic factors such as sea surface temperature and upwelling patterns. And various lines of evidence suggest that Leptospira was endemic in the sea lion population, meaning it was able to persist in the absence of introduction from some other host species or reservoir. We also learned that chronic asymptomatic shedding occurs, so individuals that can shed through time without showing any signs of infection. And through modeling work led by a former postdoc at UCLA, who's now an assistant professor at Southern Illinois University, we confirmed that this chronic shedding could be a mechanism allowing for long-term persistence of the pathogen in the sea lion population. So although outbreaks occur in the fall, and at this point most of the susceptible fuel is consumed, even if there are just a few long-term shedders, they can continue to pass the infection to new susceptible sea lions throughout the year until it gets back to the fall when conditions are good for really the bigger outbreaks, and this drives endemic circulation. And then we were very interested when, or surprised rather, when we saw that Leptospira disappeared in 2013 and didn't come back until 2017. And at first we weren't entirely convinced that it had truly disappeared. We wondered maybe it's just a less pathogenic strain and animals just weren't stranding from it, but they were still getting infected. But the 4 years of data during this fade out event, and then also the data that came from before and that followed, convinced us that indeed it had disappeared. So here what I've done is I've zoomed into that period of intensive sampling from 2008 to 2022. And in the stranding record, you can very clearly see, so the stranding record are those hills in pink. And in the stranding record, you can really clearly see that period of fade out from 2013 to 2017, which I've indicated in blue. I've also separated the older animals from the yearlings, who are essentially all susceptible as they leave the rookeries to head north. So because the yearlings are so young, they are a good indicator of what is happening with Leptospira infection in that year. And then I've also excluded the pups because to date we haven't seen any evidence that they are exposed and become infected to Leptospira. Then over the stranding record, I've added the antibody titers. And if you recall, those antibody titers indicate prior infection. And here I've plotted them in these gray circles by sampling day. And the gray circles are transparent and jittered to help with visualization. So if you see a really dark patch, that means that a lot of the little circles are overlaid and causes a darker gray. All of the animals that tested antibody negative make that thick line that you see at the bottom of the plot, because those were all negative. We plotted them as zeros. And previously we've conducted work characterizing the antibody kinetics post-infection. So we looked at the rate that the antibody titer, which is like the magnitude of the antibody response, we characterize how it declined over time. So we know that titers above a certain level, shown in this blue dotted line, are indicative of a recent or current infection, and that those that fall below the blue line are more indicative of past historic infections. We also tested animals to see if they were infected using PCR, and I've plotted infection prevalence with sample size as numbers on the figure, and then the gray bands are just the 95% confidence intervals. So a narrow interval shows that the seroprevalence estimate is quite accurate. A really wide interval suggests that it's not as accurate. Okay, so considering these different data streams, you can see that during that fade out period, despite the really huge sample sizes, there were essentially no leptospirosis strands. There were no yearlings that tested antibody positive, and the older sea lions only had low antibody titers, so no evidence of recent exposures. And there were no animals that were PCR positive, or which shows the active current infection. So given these data, we can say with confidence that Leptospira had disappeared for several years. There is one exception that I want to draw to your attention. There was this older sea lion in 2016 that had both a high antibody titer and was PCR positive, indicating a current infection. And we believe that this animal represents a failed emergence, so a reemergence. So an animal that got infected, but wasn't really able to transmit it to other animals. So an outbreak that failed to take off. And then the final nail in the coffin really comes from these phylogenetic analyses of isolates collected from sea lions from before and after reemergence. And a phylogenetic tree basically shows how related the different isolates are. And these analyses were performed by Riley Mumma, who was a graduate student at UCLA at the time and is now with USGS. And these analyses provide really nice, clear evidence that the reemergent strains were genetically distinct from the pre-fade out ones. So if you look at the tree, you can see that the pre-fade out strains, which which Riley has put in black, they form a separate group or clade from the reemergent strains, which are below them in blue. I've also included a couple strains that were collected from elephant seals. Those are in orange, and you can see that they fall within both the pre-fade out group, but also below the post-fade out group. And this suggests that the elephant seals might be exposed both by sea lions, but also maybe by other reservoirs. So why did this fade out occur? We convinced ourselves that indeed it had occurred, but now we needed to try to understand why. So first we thought about, well, what's needed to sustain an outbreak? You need susceptible animals, and you also need contact between those susceptible animals and individuals that are infected. So in order to understand what might have caused the fade out, I'm going to give you a little bit of background on sea lion contact. And I'm going to give you a crash course on the sea lion life history strategies, which might be something that you all are very familiar with. But for those of you who aren't, I'll just speed through this. And I'm going to focus on the age and sex specific differences in their spatiotemporal movement patterns, which we think really play a major role in Leptospira transmission and the seasonality of the outbreaks. So what you're looking at now is a map of the west coast of North America. And the US sea lion stock ranges from the rookeries on the Channel Islands off the coast of Southern California all the way up to the Gulf of Alaska. But there are really big differences in movement depending on the age and the sex of the sea lions. So here I've zoomed into California, where we think most of the Leptospira action is occurring. Females have their pups on the Channel Islands, or 90% of the sea lion population has their pups on the Channel Islands. There are some females that are starting to have pups up on Año Nuevo, and then even I think there are some new rookeries forming up on the Farallones and maybe farther north. But the majority of the sea lion population has their pups on the Channel Islands in May and June. And then apart from 3 to 5 day foraging trips, the females stay on the islands to nurse their pups for around 9 to 11 months. During the same time period, essentially the entire population returns to the islands for the breeding season. And that's April, May, June. So from a disease ecology perspective, this is a well-mixed population, meaning all the different individuals are kind of contacting each other within the population. However, outside of the breeding season, all but the moms and the pups, or essentially all but the moms and the pups, head north, with the older males leaving later and going further north. Animals continue to forage in the spring. The pups are weaned, they leave the rookeries and start to travel up the coast. And then with summer, everyone heads back to the islands and the cycle begins again. But I want to back up to the spring, which is where we believe that a crucial link between the fall outbreaks occurs, when the yearling population, which our work indicates is essentially entirely susceptible initially, when that yearling population begins to migrate north and encounters infected older animals along the coast. And when that happens, we think the necessary spark gets thrown into this new pile of fuel, and then we see an outbreak. So we were thinking about these age and sex specific movement patterns when we were trying to figure out why fade out may have occurred. And thanks to the long-term data sets on sea lion demography and movement that I mentioned that our Noyo collaborators have collected, and through conversations with these colleagues, we were able to look for anomalies in demography and movement as possible explanations for the fade out. And so what I've plotted here is on the y-axis, you can see the proportion of yearlings that leave the rookeries in the spring. And on the x-axis, the population of the size of the yearling population in thousands. And it turns out that in 2013, which is the year that we saw fade out, the yearlings never left the rookeries. So there wasn't any fuel for a spark to fall on and cause a fire. In addition, the on the y-axis, so here you can see that on the y-axis, the proportion of yearlings leaving the rookery was quite low as they were weaning, as they were weaned and they were leaving the rookeries in the spring. The black dots are the pre-fade out years, and whereas in years prior to 2013, which are all these years in black, the majority of the yearlings left the islands. In 2013, you can see that they stayed. In addition, the survival of the 2012 pup cohort, which is the 2013 yearling cohort, their survival was really terrible. And as a result, the cohort itself, there were very few of those animals. So the cohort was small, and you can see this looking at the x-axis. However, you may note that there are other years with similarly small yearling populations when they still went north and outbreaks occurred. But in 2012, not only was pup survival really quite poor, but so was pup growth. So those 2013 yearlings themselves were quite small. So not just the population size in general, but the individuals were quite small. And you can see this on the x-axis of the right-hand plot, where the pre-migration weight in 2013 was much lower than what you're seeing in any of those other years. And we think that was what kept them from leaving the rookery. So finally, we also looked at the stranding data from the Marine Mammal Center, and it shows that of the yearlings that did leave the rookeries, very few actually traveled north where they could have encountered these older infected animals. And you can see that on the plot to the far right, where I have month plotted on the x-axis, and the y-axis is the proportion stranding above 36 latitude, which is about Monterey Bay. You can see that in the map of California in the bottom right. And in 2013, very few of the yearlings were stranding above that 36-degree latitude line, whereas in other years, many of the animals that stranded above that line. So basically, this shows that of the animals that were leaving the rookeries, very few went north. So we believe that the crucial link between the fall outbreaks, when susceptible yearlings leave the rookeries and encounter older infected sea lions further north, just didn't occur in 2013, and that the transmission chain was broken. Digging a little deeper, we learned that there had been a lack of sufficient high-quality prey in 2012, 2013. And the left-hand figure shows a declining anchovy and sardine abundances in the adult female foraging range. And those are two very prime prey sources for the sea lions. And then the figures on the right show a decrease in the frequency of occurrence of these same species in adult female scat, shown by the gray bars, and how that was associated with a decrease in pup weights, which is shown by the black lines. So essentially, the anchovy and sardine populations crashed. The females weren't getting sufficient high-quality prey, and this significantly impacted growth and survival of the 2012 pups that then became the 2013 yearlings. So the obvious next step in a time of unprecedented oceanographic conditions and climate change was to try to understand whether climate was linked to these events. So I've spent a significant amount of time reviewing the literature and talking to experts. And as it turns out, it's complicated. So even the fisheries experts were struggling to find consistent, clear climate drivers of forage fish abundance. Seidemann and colleagues were able to find at least one relatively clear relationship, and you can see this up in the top right corner. They showed that over the last 70 years, anchovy biomass in the foraging range of adult female sea lions has generally tracked the spring upwelling index. However, this relationship, as is the case with many others that had historically held true, has broken down over the last 5 to 10 years of really unprecedented oceanographic anomalies and climate change. Okay, so I've explained why Leptospira disappeared. The next obvious question is, why did it come back? And interestingly, it appears that more than just a return to favorable conditions was needed for reemergence. Here you can see in 2014 that although the yearlings were in good condition and were leaving the rookeries, there was no outbreak. So we kind of scratched our head and wondered what was missing. And we also saw we were thinking, okay, so the condition was good, they're in the right place, but you also need to have a spark to light the fire and start the spillover, start the outbreak. And we think that this sort of spark or spillover event occurred in 2016, when we saw that single case of leptospirosis at the Marine Mammal Center that I mentioned earlier. But this time, when there was that spark, the conditions were terrible. There weren't many yearlings, and those yearlings that existed were small, and they didn't leave the rookeries. But then in 2017, the conditions were great. There were a lot of yearlings around. Their pre-migration weight was great, and they left the rookeries. You can see that they, from the stranding record, that they also were stranding above the 36 latitude line, and so they were migrating north after they left the rookeries. But we were left scratching our heads because the size of the 2017 outbreak was much smaller than we would have predicted based on epidemiological theory. So you would imagine that after a 4-year period of no outbreaks, the susceptible population would be enormous, and that as a result, you would have just a huge outbreak. But instead, we had a relatively small outbreak, and it wasn't until 2019 that we saw a big outbreak. And when we dug a little deeper, we saw that not only was the outbreak small, but it started late, and the age distribution was atypical. So here I've plotted, at the very top is the 2017 outbreak, the middle is the 2018 outbreak, and the bottom is the combination of all outbreaks that occurred before Fadeout. And the ages are stacked with the youngest on the bottom and the oldest on top. So the red and orange are the younger age classes, and the green and blue are the older ones. And once we looked at the overall population age structure shown on the right, we realized that in 2017, the outbreak was strange because during those fade-out years, pup production and survival was generally pretty low. And in 2017, you can see here on this lower right-hand corner, there just weren't many young animals around to get infected, even though they were going to the right place. But then by 2018, there was an enormous cohort of the younger animals, and so plenty of fuel for the fire. Okay, so to summarize what I've just gone over with the sea lions, the outbreaks are sparked in the spring when the yearlings go north. Poor foraging conditions, which we believe were driven by oceanographic anomalies, led to really small yearlings. Those yearlings ended up staying on the rookeries, which instead of going north, which is what they typically do, and we think that this broke the transmission chain and led to the Leptospira fade out. And then Leptospira returned when the conditions, when the necessary conditions were met. There were sufficient susceptible yearlings. These yearlings traveled north, and there must have been a spillover event to spark an outbreak. So then the next logical question is, where did this spillover come from? there's a prior grad student, now a postdoc in the lab, who has been working to collect urine and kidneys from a variety of terrestrial animals to look to see if they have Leptospira infections, and then also to get isolates for our phylogeny. And we're working on that right now to see where they fall in the phylogeny. But then we also are curious about what other marine mammals what their roles might be in the transmission chain. And we know that northern fur seals, harbor seals, stellar sea lions. Recently, we saw a case in a short-beaked common dolphin. Northern elephant seals and sea otters can all get infected with Leptospira, but we don't know if they're all getting infected with the same strains that the sea lions have. I have some more slides talking about looking for evidence in elephant seals, but that is probably another 10 minutes, and it looks like we're kind of getting close to 7. So maybe I'll just summarize it super quickly by saying that we looked at antibodies in northern elephant seals during that fade out period, and didn't see any evidence that northern elephant seals were maintaining Leptospira during that fade out period. So it's unlikely that elephant seals are the reservoir and the source of spillover to the sea lions post fade out. But it does seem that they do routinely get infected. But as I had mentioned before, if you look at the phylogeny, the elephant seals seem to get strains that are similar both to the California sea lions pre-fade out, but also strains that aren't related to any of the ones that we've seen in sea lions, suggesting that they might get infected sometimes by sea lions, and they might also sometimes get infected by other species. So it's possible that elephant seals could have been what we call a bridge host, where maybe they were infected by some unknown species, and then they infected the sea lions, and that started and that might have started the outbreak during reemergence. We also recently have some new collaborators with some fancy new tools. We traditionally had to grow cultures of Leptospira, which is very difficult and has to be done with fresh kidneys in order to be able to build these phylogenies. And now these collaborators at Northern Arizona University have this great approach where they can get whole genome sequences or close to whole genome sequences from archived kidneys. And so we're really excited about that. And we're currently writing grants to get funding to be able to get more sequences using this technique. The only problem is it's about $2,000 to $3,000 per sample to use this technique, and so we have to apply for a bunch of grants to be able to do these analyses. But we're really excited to run these analyses on non-sea lion samples. So we have a number of samples from elephant seals and harbor seals, otters, northern fur seals, stellar sea lions that are suspicious Leptospira cases that we will test by PCR, and then if they're PCR positive, we will try to sequence them and put them on our phylogeny, because that will be really important for us to understand how the different populations are epidemiologically linked, and how Leptospira is transmitted across the Eastern Pacific marine ecosystem. So again, I'd just like to make sure I say a huge thanks and mention all the really wonderful collaborators that I have, and then also acknowledge all our various funders and the permits under which we do this work. And I would be happy to take any questions that people might have.
I have a question.
Sure.
So is this something that's been going on for a long time and we're just learning more and more about it, or is this a relatively new thing? Is-
Do you mean the leptospirosis in the sea lions or
Yes,
leptospirosis in general?
in the sea lions, because that's sort of what you're specializing in.
Yeah, so the first recorded outbreak of leptospirosis in California sea lions was in 1970,
Oh,
when sea lions were stranding up and down the California, Oregon, and Washington coast, and they isolated Leptospira interrogans serovar pomona. And then um, the the data from the Marine Mammal Center didn't really the data from the Marine Mammal Center doesn't really show many cases of or any cases of Leptospira again until around 1983. And we don't know if that's because at the same time the sea lion population was relatively small, stranding and rehab effort was relatively low, or if maybe there was a big outbreak in 1970 and then it faded out and then came back and then at that point has been circulating consistently since 1983. And it it has had an impact on the overall sea lion population in that our NOAA collaborators have looked to see associations between big outbreaks of Leptospira and um, any noticeable changes in the population size. And it has, in the really big outbreak years, you do see a reduction in the kind of juvenile male age class. But despite the fact that there have been yearly outbreaks of Leptospira since the 80s, the population has continued to increase
oh,
through
that's
that time
good.
period. So in the 70s, the sea lion population, I believe, was around 30,000, and now it's around 300,000.
Wow.
Yeah.
So I noticed before this, I read, I mean, basically, I was reading a little bit about this bacteria, and it's all kinds of variations that infect all kinds of species throughout the world. So I'm just amazed. I didn't know anything about this
Yeah. So
bacteria before that.
yeah, so we don't see it much in the United States,
Ah,
in humans. If you have a dog, you probably get the Leptospira vaccine, but it's only against 4 serovars, and there are over 300 pathogenic serovars. There is cross-reactivity, but how protective that vaccine is still kind of a little bit up in the air. They think that they've picked the most important serovars um, for for what the dog risk is, but we do see leptospira infections in a lot of wildlife. But because it's transmitted through contact with those infectious leptospiras, which are shed in urine, unless you're coming into contact with the urine of infected animals, or if your mucous membranes or abraded skin is coming in contact with contaminated soil water, you're not going to get it. So like you tend to see it in more developing countries where maybe there are the rodent hosts that might be foraging in rice fields or something.
hmm.
And when people are harvesting the rice, they might end up having scrapes and coming in contact with the urine. Or there are tons of cases of it in Brazil. Um, and that's because the main reservoir is the rat, and
Oh,
there isn't good sewage infrastructure. So if there's flooding, the sewage all floods with it. People have to fix the sewage, and they come in contact with
hmm.
the infectious leptospires from the rat urine.
So another question, if you don't mind.
Yeah.
So does this, no, I'm not sure. You were just mentioning it as a virus. I thought it was a bacteria. So
No, did I say virus? It's a spirochete bacteria.
okay.
Yeah.
Oh, that makes sense.
Yeah.
And does this infection change the behavior of the animals a lot?
Um, it's not like toxoplasma where they think it, reduces fear in the rodent host. It 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 changes the behavior in that if they are clinically impacted by it, they just feel crummy, so they're going to be lethargic. They're going to have a painful abdomen. They're going to be kind of depressed.
Well,
And
what I meant was, like there's some infections to things like ants that make them want to crawl up to the top of things, and that
yeah,
causes the infection to be spread by the fact that the ant explodes when it gets to the top of a tree or a bush or
yeah.
something. I was thinking more on those lines. So, as far as you know, nothing like that
Well, I
extreme.
mean, it's shed in the urine and it
Uh-huh.
causes damage to the kidneys, and the damage to the kidneys makes them thirsty, so it makes
Right.
them drink more and urinate more. So in that sense, I guess, if they're urinating more, they're potentially shedding more of the
Right.
infectious leptospires, but nothing like those weird infected ants and stuff like that.
No, I didn't think it would be, but I just thought that could be a possible.
Yeah, now I mean, pathogens are interesting. They do all sorts of weird and fascinating things.
For sure. Yeah. Thanks, Bruce. Those are good questions. So I obviously dog owners are concerned, and I think that that's one of the most frightening things up here when we do have a lepto outbreak like we did
Mm,
this year, just for in terms of understanding the lepto connection for canines that are walking along the beach and coming across a dead sea lion, or walking along a beach and rolling where a sea lion just peed, or all those potentials. And then even the vaccines that we're getting with all the different strains of lepto, how easy is it for us to understand which lepto vaccine are you going to give to your dog? And do you
yeah.
want to dive into that just a little bit for
Sure,
our?
yeah. So the vaccine I think is a 4 serovar vaccine. And there's really only kind of one vaccine that you can get. So you're just gonna get whatever the lepto vaccine is. I would definitely recommend vaccinating. I don't think Pomona is actually one of the serovars that's in the vaccine, but I think there's probably cross reaction to Pomona, which means that there's probably some protection against it. I would definitely recommend keeping dogs away from any sick sea lions for a number of reasons, but one of them is because of lepto transmission. And similarly, like if they have any abrasions on their paws and they walk through sea lion urine, it's possible that they could get infected. That all said, I would love to get isolates from infected dogs. And I know that we've had some like little outbreaks of Leptospira in domestic dog populations. There was one down here in LA, but it turns out it was Leptospira interrogans serovar canicola, which is the one that dogs tend to be the reservoir host for. And it was I think it was in shelter dogs.
Mm, yeah.
Um, and so I would vaccinate, and I would keep your dogs away from dead marine mammals. And if there's a big lepto outbreak year, I would avoid beaches where a lot of sea lions tend to hang out.
Right. No,
Yeah.
that's great information. Thank you.
Yeah.
And then for the other animals, marine mammals species, do they have the similar leptospirosis symptoms? And do they have different susceptible populations that you know of?
Yeah, that's a really great question. The short answer is we really don't know much about the other populations. There's very little published. Um, I I know for a fact that elephant seals can get ill from it. We have seen cases at the Marine Mammal Center of elephant seals that are sick and have died. Some have also survived. They're usually the weaners, and because you don't really see anything much older than a wiener. And then because of the way the natural history is for the elephant seals, it's hard to know whether the older animals get sick and die because they're pelagic. You know, they take off into the deep ocean for months at a time. And so not only would it be hard to detect them getting infected, like if they died, they're just kind of floating out there, but also because of that life history pattern, they don't have a lot of opportunity to get infected. But when I did a study looking at infection in those elephant seals, and here is in the line is the seroprevalence. The dots are the actual titer, and then it's broken down by pup, yearling, and animals older than 2. And you can see that there's a decent proportion of the animals that were tested that had antibodies. So we do see evidence of infection in elephant seals, and we know that they can get sick from it. In terms of the other species, I have a friend and colleague who is the pathologist for California Department of Fish and Wildlife. And she's done a lot of necropsies on sea otters that she thinks are suspicious of leptospirosis, but and their kidneys are on hold for us to test if we can get the funding to do that. Harbor seals, there have been harbor seals that have died from Leptospira. It's not common. And we definitely have seen antibodies in harbor seals, but the California sea lions are the only ones that we see stranding in these huge numbers due to Leptospira. We know that stellar sea lions can get infected, and I believe they show clinical signs. And then there's only this one case of the short-beaked common dolphin, which is the first cetacean in the northeastern Pacific to have been reported and tested positive for Leptospira. I have a colleague who works in Brazil, and he has detected it in a number of other cetaceans and is publishing that work now. But those have all been animals that died, and he doesn't know what the cause of death was on those. So whether or not it was causing clinical disease in those animals, I'm not sure, or we just don't know. Yeah.
Yeah, and then Catherine Keegan has an interesting question. It'd be interesting to know if the infection makes seals and sea lions more likely to be preyed on by great white sharks. Seems likely, but difficult to get that kind of information. But just doing rescue, when you see these animals that are sick with leptospirosis, they if they were out there, yeah, they'd be pretty easy for them to get picked off, and that's kind of why they come ashore. Because they're just feeling crappy. And when you see a lepto animal, when you see a little sea lion who's got lepto or a big sea lion, it's kind of brutal to watch. They're just that lepto tuck, and we never diagnose in the field because people will laugh at us, but you can see these animals. Even we had a dead one this year that when we got like 10 animals, they were all subadults, by the way. They were all, because up here we don't get the little guys very often.
Right.
But everyone was between like 150 to 200 centimeters. And above 200 centimeters, we didn't see anybody. So it was a subadult cohort that came in pretty fast and furious. Within a few week period, we had all these subadult sea lions that were dying. And then as well, we were rescuing a bunch that were being sent to the center that subsequently died. So it was this interesting, really concentrated little cohort. And I'm sure the Marine Mammal Center numbers will paint that picture on a larger scale. But for us up here, it was kind of like, whoa, what's going on? Every other day or something, we were either rescuing or responding. And that's when I started grabbing little kidney samples for you. But
I love to hear that.
That's how we
that makes a lot of sense. because, the bigger guys are the ones that are up here where you live. The little guys don't really go that far north. And so it makes sense that as they're going north, they might kind of be bringing the outbreak. And the peak of lepto outbreaks tends to be in October. So if it's kind of peaking around the Bay Area, maybe it takes a little bit longer to make its way up your way. And certainly my colleagues in Oregon and Washington, it seems to me, and it's hard because the data aren't as, they are not able to do as many diagnostics in Oregon and Washington because most of their animals are found dead. But it seems like they typically see them later in the year too.
Mhm.
Yeah.
Yeah. And we wondered too if that was actually kind of a cohort that was susceptible, that like didn't get it during 2013, 2014, and then I don't know that this, well, that wouldn't make sense, but if it was a cohort that was, just this bunch at this age
Yeah.
class that are getting it.
So the puppies born in 2017 would have been part of the 2018 outbreak, which was really huge. And
Mhm.
so those animals, if they were born in 2017, and you're saying it's 2022, they
Right.
would have been 5,
Mhm.
So the 4-year-olds are the ones that were born. So the ones born in 2018 that were puppies in 2018 wouldn't have been exposed because for some reason the pups aren't exposed on the rookeries. So I guess if the animals you were seeing were mostly 4-year-olds, 3 and 4-year-olds, that would make sense.
Yeah, it'll be interesting to see those numbers, and just
Yeah.
see what was going on there. Let's see. I want to make sure I get to everybody's.
Oh yeah, Sarah. Let's see.
Go ahead.
I think the next question is Sarah Bogard.
Yeah, go ahead.
Yeah. So that's an excellent question about how long it survives in soil and sand. So the answer is not simple. Nothing
I would
about
be
Leptospira is simple.
I would be surprised if it was simple.
Yeah, it really depends on a lot of different factors. So traditionally, it's been believed that it doesn't survive well in salt water. So it's been kind of a puzzle about how a marine mammal can be transmitting it, but sea lions spend a lot of time on land. We also have done some unpublished work that shows that it probably can remain infectious and viable for like 6 hours. So when sea lions raft and urinate and defecate in those rafts, then it's possible they could be transmitting it there. In moist soil, lepto can survive for months, and there's some work that has shown that that after a big rain, like it binds to the substrate, and then after a big rain, it resuspends into the water, and so you can have lepto kind of accumulating in the soil, and then if you get a big rain, it comes up, and then that can lead to transmission events that can occur associated with rain. Sand, it seems like I don't remember the data on sand, or if there are any data on how long it survives in sand, but yeah, it can be really variable. I think probably like with sand, it probably, since it's so porous, the urine probably just goes down. But that is one of the big questions we have is how is it being transmitted among the sea lions?
Because they crawl all over each other and pee all over each other.
Anybody who's watched them is like, of course it's pretty easy. Like
harbor seals are like, no, get away, dude.
Exactly.
Don't pee in
Yeah.
this.
And like, they come out on these rocky outcroppings, they pee on the outcroppings, they scrape their flippers, then they have these open wounds that they can then pick up the leptospire. So we don't have we're not able to show definitively how it's transmitted, but I mean, there are a lot of really viable theories.
Sure. Let's see. Everybody's just really fascinated and loves
Oh, great.
shared for sure. I had just one question. We had 3 animals in this area that had green tags.
Oh, those are mine.
I thought so. That's what I think Sue Pemberton mentioned, that those were your tags.
Yeah.
So we had one little youngster, I think he was probably 3-ish, and we tried to get a hold of him, and he was in that leptocoral, or in that time frame.
Huh.
And like, yeah, 3 or 4-year-old, but he never stranded in a location where we could get to him. And then he was just so thin and so sad looking and then he disappeared. And then the other one was a big guy on the dock who looked to be in good shape. Adult, subadult, hard to tell.
Yeah.
And then the last one was a really severely shark bitten animal that was, again, probably subadult, adult age. So those three sightings with that. So just these double green tags. So folks, when you're there and you happen to see a bunch of sea lions on the docks at Noyo Harbor or wherever you are, if you get the picture, even if you don't get the number on the tag, it's kind of cool to get get those double greens. And I think one of the youngster was 377.
Oh, okay. Yeah. Well, if you ever see them and you get a definitive tag number, let me know and I can look them up and tell you when we caught them and any of the associated information. And we love to know about those resightings because it's always nice to know what's going on with them, but also to know that our handling actually wasn't harmful to them, and that they continued to survive for years, and then we'll potentially sadly got bitten by a shark or whatever. But
he was amazing. He was bit just really bad on one flipper, but he had figured out how to move and he actually was gone the next day. So I was like,
it's amazing, the shark bite survivals.
Yeah, very cool. So yeah, I have the numbers on two of those tags. I think he was 229. So I'll send you pictures and locations from those. And let's see, did we have any other questions? Yes.
Yeah, there
I
was
have a simple question.
Hold on, let's get to the chat, folks, please.
Yeah, it looks like Alice had some questions about the oceanographic conditions and changes in fish populations during reemergence. And the short answer is yes, there were changes. You can see some of these evident through 17 and 18 differences in the sardine and the northern anchovy numbers. And there's just been a lot of kind of chaos in oceanographic factors over the last decade or so. Like at first we were thinking, the fade out, it aligns with the blob, but really the fade out happened at early 2013 and the blob kind of impacted Northern California starting end of 2013, early 2014. But the blob was through to like 2015 or so, and then there have since been other marine heat waves, and so there's just a lot of flux right now. So it's hard to predict things. Yeah.
Would you say that? I mean, I feel like with the warming, are El Nino years, do they tend to be bad years, or is that something from?
Yeah, it's an excellent question and one that everybody has asked, and there is no association with the El Nino years. Because I think again, I keep saying this, it's kind of a cop out, but it's really complicated. And so, there are so many different factors. So you've got different ages and sexes of sea lions in different places along the coast, and that's going to impact their mixing dynamics. They all have different diets. And then the forage fish populations also have really different age specific needs and distributions. So like, the young of the year or like the spawn end up like needing certain conditions. And there was one year where because of the way the um, winds were blowing, all the spawn got blown offshore, and so they didn't survive well. But like the temperature conditions were such that you would have expected them to. So it's all like all these little nuances, and then you go 100 miles up the coast, and it's totally different. And you're like, how am I supposed to like organize all this? So when we figured this story out with the Fadeout and the reemergence, we were shocked that we could come up with this nice of a kind of tidy story. Yeah.
Thank you.
Thanks so much. This was really fun. I love what I do. I think I love this story. I think it's really interesting. So thanks for the opportunity to yammer away about it for a while.
100%. So great. Well, hey, thanks so much. We really
thank you.
Come and visit us if you're ever on the North Coast.
Yeah.
Thanks so much, everyone. Have a lovely rest of your evening. Thanks again,
everybody.
Thank you.